Today we're going to talk about nucleic acids. We're going to talk about their structure. So first of all, we're going to learn about 5 things. Difference between RNA and DNA. We're going to talk about the similarities between RNA and DNA. We're going to talk about the central dogma of life, what that is, and we got to understand their structures. You'll recognize and draw structures, and we want to predict complementary base pair for DNA with a messenger RNA or DNA with a DNA. So those are our learning objectives. So what are nucleic acids? They're substituted polymers, biopolymers, and they have an aldo pentose sugar, and they carry the organism's genetic information. This is page 1242 to 1249 in your textbook. So if we made a little chart, and I would recommend you do this, RNA, DNA. So that's ribonucleic acid. DNA is deoxyribonucleic acid. This is mostly found in the cytoplasm of the cell. This is found in the nucleus of the cell. This is much smaller than DNA. This is really a huge molecule, molecular weight about 50 billion. We also have messenger RNA, transfer RNA, and ribosomal RNA. So you need to know those 3 types of RNA. They're usually single-stranded. And I say usually. DNA comes double-stranded. So that's going to look like this. In fact, let's make one of them red. Okay. And then we'll make the other one blue. Okay. And then you're going to find that they base pair. And we'll talk about that, what that looks like. One strand is going to be from the 5' to the 3', whereas the other would be from 3' to 5'. This is the anti-parallel complementary base pairing. So that's your secondary structure. This is secondary structure. And we'll also talk about the primary structure. Primary structure is going to be the structure of your nucleosides and nucleotides. So the nucleotide consists of 3 things. You have the phosphate, you have the sugar, and you have the base, the N-heterocyclic base. The nucleosides are 2 things. The sugar plus your N-heterocyclic base. DNA is more stable. It's harder to break down and degrade. And the difference here for RNA, you have your sugar, okay, is going to be a ribose sugar. So we're going to look at what that structure is. And your sugar for DNA is going to be deoxy, 2' deoxy ribose. And another difference for your in heterocyclic bases, you have uracil in RNA, and you have thymine in DNA. Okay, so there's going to be these two structural differences. And so let's just look at the overall central dogma. So inside your nucleus, you have your DNA, and it's double, it's a double helix, and it's in the nucleus. It's large. It's in the nucleus of the cell. Okay, so if this is your cell, this is your cytoplasm, these strands will come apart with the help of enzymes, and you get these pairs here. Okay, these are called daughter strands, and each one contains the genetic information. Okay, so what's on these here, these little black marks that I'm making here, are the bases. So those are the bases in heterocyclic bases. And we're going to talk about what those are structurally. And we have for DNA, you can have A, G, C, or T. So you can have adenosine, you can have guanosine, you can have cytidine, well, cytosine, and you can have thymine. Okay. Now, if it's RNA, you have adenosine, you have guanosine, you have cytidine, but you have a uracil. And here you have a thymine DNA. Okay? So those bases, what happens is messenger RNA, so we have a messenger RNA, which is single-stranded, SS, single-stranded, can come into the cell and it can double bond or make the double helix here. Okay, and then it can come out into the cytoplasm, and what it does then is it base pairs and then it comes out. Okay, so then it comes out here. Okay, and then so it gets the information. Okay, so now it has the information and it binds to an rRNA, ribosomal RNA. You'll learn a lot more about this in biochemistry next year. And then you have transfer RNA, and they kind of look like this. They have a weird shape here, and they have an amino acid here, and they have an RNA here. And transfer RNA comes in and will bind on the ribosomal RNA, so it'll interact with, you have messenger RNA, you have ribosomal RNA, and then you have transfer RNA, all with this complex. And then what happens is these amino acids come off, amino acid 1, and then you could have another transfer RNA. These are tRNA. They have another amino acid, and then it bonds to make an amino acid 2, and this makes a peptide bond. And these are your proteins. Okay, and your proteins are your workers of the cell. These are the workers. So the central dogma of life is double-stranded DNA, then goes to messenger RNA. Okay, this is called transcription. Transcription, I believe. Always have to double check those. So I'm going to look in your slides real quick. And I'm wondering, does it include in your slides? Let's see real quick. So you can always get your phone out. Transcription. Okay. And DNA. Yeah, so transcription is going from DNA, transcription, from DNA to a messenger RNA, and then messenger RNA combines with ribosomal RNA and transfer RNA to make proteins. Okay, and this is called translation. And these are the processes to make proteins. And so this chapter, we're really focusing on the structures of the nucleic acids, which are RNA and DNA. So let's look at the structures. Okay, so if you look at sugars, we've looked at the Fisher projections. And so what does ribose look like? Let's draw ribose. Ribose has an aldehyde, and then you have your OH here at 2. You have another OH here at carbon number 3. You have OH here at 4, and then you have CH2OH. Okay, so this is the acyclic form of ribose. Now, in the body, it's in the Haworth projection. And the Haworth projection is the cyclic. So equilibrium, this is probably 99% cyclic with maybe a 1% in the acyclic. And so what happens is, so let's number this, 1, 2, 3, 4, 5 carbons. It is the 4th carbon here, this oxygen, that will nucleophilically attack that carbonyl and will make a hemiacetal. So when you do that, the number 4 oxygen is here, and you've probably seen this structure before, and this oxygen, then this carbon is here, and let's just go ahead and do that. Carbon number, so that's 1, this is 2, this is 3, this is 4, that's carbon number 4, and then 5 is the CH2OH. And so this is a ribose sugar, and it's closed like that. Now, the bases that you have are the N-heterocyclic bases, and they look like this. Now, these are called pyrimidines if they're just a single structure, because pyrimidine, the actual structure of pyrimidine is this right here. Okay, this is actually pyrimidine. And so now if we just draw this structure with our nitrogens, all right, and this would be, this is the nitrogen which is connected to the sugar. Okay, this is cytosine, and cytosine if it's just the N-heterocyclic base, and this is uracil. Okay, yeah. Now, if it's attached to the sugar here, so let's redraw this sugar of ribose, and let's put on the uracil. Okay, now this, instead of uracil, this is uridine. Okay, I think a D, I for 2, and I think of it as the N-heterocyclic base plus the sugar. Okay, so you see we have this bond here. This here is called the glycosidic bond. And this is very important. This is where your bases are. So when you have your double-stranded DNA strand, these bases do a pi stacking, okay, and they're bonding over here, so they're actually like at an angle bonding. Okay, now, because these get the number 1, 2, 3, 4, 5, 6, the sugar gets the 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, so that's how the numbering goes. And then if you want the nucleotide, then you would put the phosphate on here. And so you would put a phosphate. Phosphorus likes 5 bonds. Okay, let's get this right. And you see how it's negatively charged. So when you draw this out here, the phosphates are all back here. Okay, this is the phosphates. It's on the outside, and they like to form, they're water soluble. Okay, this portion here, the bases, then would be inside, and so that's how you get your secondary structure. So let's look at this again. These are RNA. Okay, so because RNA, the R stands for ribose. So ribose nucleic acid. So in the DNA structure, we have deoxyribose nucleic acid. And this is a 2 prime. So what's different here is our sugar structure would be such as this. Okay, so then you have 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And the DE in organic chemistry means without. So this is without an oxygen at the 2 prime carbon. And that is your DNA sugar. So it's a deoxyribose sugar, and that's a big difference. And this makes it more stable. So DNA is a lot more stable to resistance. And the other difference is the base. So let's draw the base of thymine. Um, so we draw our pyrimidine here, which is a single structure there, and you get your CH3 here. Okay, so this is thymine, but when it's added with the deoxyribose, it's called thymidine. Okay, and then if you add the 5 prime hydroxy, 5 prime monophosphate, then you have your nucleotide. Nucleotide is 3 things. So that's the difference in the structures. What they do have in common are the purines. Okay, so continuing on with the structures, what's in common is the purines, adenine, and guanine. Okay, so they're the, and so what does a purine look like? A purine looks like this. And nitrogen, we can do a better structure than that. Okay, this is a 5-membered. Okay, so here we go. We got 4 bonds. This is a purine. Okay, and you're going to see the connection to the sugar at this carb, at this nitrogen. Okay, so we have, so if you draw that framework, draw your 6-membered ring, your 5-membered ring, drawing your nitrogens, and then for adenine, this has an NH2 here, and then you do 4 bonds. Okay, these are purines. I think a bigger structure, smaller word. That's how I keep them straight. Okay, the other one is guanine. And so let's draw our 6-membered ring and our 5-membered ring and put our nitrogens in. And then this has, see how this has 4 double bonds? Not carbon double bonds, but just 4 double bonds. Okay, so there's 4. This one has 3. So you get a carbonyl up here, you get an NH2 here, and so there's a double bond, there's a double bond, there's a double bond, and at the nitrogen is where you get connection. Okay, so those are the difference in the structures. The other thing I need you to be able to do, our 5th one, is predict the complementary base pair. Base pair. Okay, so purines will base pair with the pyrimidines. Okay, so A will base pair with the T, and it does 2 bonds, 2 hydrogen bonds. And you know that that's going to be in DNA. Whereas adenine will 2 base hydrogen bonds with uracil and RNA. And then and G will do 3 hydrogen bonds with cytosine in both RNA and DNA. Okay, and where do those hydrogen bonds happen? You should look at that in your book. Adenine, let's draw this with your typical, let's do, okay, so obviously this is uracil. Okay, so hydrogen bond here between this hydrogen and this oxygen, there's 1, and then between this nitrogen and this hydrogen is your double bond there. Guanine with cytosine, so let's draw a cytosine here. Cytosine, nope, that's not cytosine. Cytosine has this NH2 at the top, and there we have cytosine. Okay, so we have 3 hydrogen bonds. This hydrogen here will hydrogen bond with that oxygen. This nitrogen will hydrogen bond with this hydrogen here, and then this NH2 will hydrogen bond with this oxygen here. So look that up, and kind of understand that structure. Understand the difference in the structures. Also, yes, one more thing. If you have DNA, so it does anti-parallel, so two complementary strands will hydrogen bond in anti-parallel fashion. So if you were given a 5' DNA, okay, so you have A, G, C, T, A, G, 3', could you predict the complementary DNA? Okay, so it would be 5' to 3', and then A does what? A in DNA would be double bonded to G, hydrogen bond. G would be C, triple bond. C would be G. T would be A, double bond. A would be T. And then C would be G, triple bond. Okay? And what if this was DNA here? 5' A, G, C, T, A, C, 3'. This is your DNA, and you want a messenger RNA complementary. Well, you would still do 5' to 3', and then A would be not T, but U, double bond. G would be C, triple bond. C would be G, hydrogen bond. T would be A would be U, uracil, and C would be G. Okay, and that wraps up what I expect you to know about nucleic acid structures.